now be able to see my CEAT slide. Okay, so for the first session, then we're going to concentrate on CEAT, which is a large collaborative project that we've been running under the banner of SMARTExpertise. And CEAT is the cluster for Epigenomics and Antibody Drug Conjugate Therapeutics. My name is Lewis. I'm an associate professor in the RBGO and co-PI on CEAT with Daya. Okay, so what's the motivation for industry academic partnerships? What are we trying to achieve? What are the challenges? And for me, I touched on it in terms of the output of RBGO and what we're trying to achieve, and it's those clinical challenges. And advancing disease understanding is a huge challenge, because if we understand the disease and we understand the inherent properties and characteristics of disease and can build models, it allows us to translate that understanding to new diagnostics, to new therapies. And it's without that understanding, we're not going to be able to add value to patients and contribute to industry development, to clinical development, and to patient health. So our motivation for CEAT particularly is gynae-oncology, and our target disease is ovarian cancer, which is the most lethal gynecological cancer. And ovarian cancer is a solid tumor. It exists primary site at the ovary, but then leaves the ovary quite quickly and expands into the local environment, into the peritoneal space. It induces a lot of lymph node leakage and development of ascites, which is this toxic fluid, and then harnesses other cells in that environment to really become an aggressive, difficult disease to treat, to detect, and to provide patient care. So our challenge, our motivation is to understand this disease, understand the tumor microenvironment in much more depth, so that we can develop new diagnostics and new drugs. So we do understand ovarian cancer. It's very heterogeneous. It's got multiple subtypes. It presents at different stages at diagnosis. We can detect at stage 1, and the survival rate is very high in the bottom left-hand corner. But we can also, the majority of patients we detect at later stage, we diagnose at later stage. And you can see there that the 5-year survival rate is particularly low in those later stages of diagnosis. And that's because it's asymptomatic, it's very difficult to detect. And when it gets to those later stages of disease, it's very, very aggressive, very metastatic. And has a particularly high burden on those patients. So even though we know a lot and we can characterize and subdivide the different subtypes of ovarian cancer, it's still a hugely challenging environment and a challenging disease for our clinical partners. And the ultimate kind of evidence of that is this 5-year survival rate, which is very low at advanced stage disease. Okay, so the first line treatment for ovarian cancer, chemotherapy and debulking stage surgery, is usually successful. It hasn't changed in many years, but because of this heterogeneity and because of this, the relapse rate is very high. So when the disease comes back, it's associated with resistance, associated with difficulty in treating, and therefore there's a need for new disease understanding, diagnostics, and advanced therapeutics. So that's really our motivation. How do we develop new understanding? How do we develop new approaches to try and manage this difficult disease? And we can't do this as an academic group. If we want to get new tools out, towards the NHS, towards patient, towards bedside, we need to develop partnerships in order to do that. So the motivation for CEAT, we sat down at the beginning of the project and in conceptualizing the project, and we wanted to identify potential epigenetic processes that may be driving cancer development. We wanted to characterize kind of biophysical and characterize the local environment of the disease to see if we can learn about the local environment and understand how we can manipulate it to try and confine ovarian cancer to its local environment, stop this aggressive metastatic spread, and really understand what are the properties and the characteristics of that disease microenvironment that allows us to target drugs to it, and to understand how we may be able to get a higher concentration of drug to those environments and target individual cell types within that, with existing drugs and new drugs. And one of the ways in which you could target the tumor microenvironment and individual cells is by understanding what proteins are upregulated in that environment, and then deriving antibodies and conjugating drugs to those antibodies to get particularly targeted delivery. Now, to do that, there's a huge number of challenges, okay? And in order to do that, we conceptualized the project with our industrial partners. And you can see here, there's a nice diagram to show how CEAT fits together, okay? So we have 7 partners. We're trying to understand different gene regulation, different patterns of gene regulation through epigenomics, trying to see if we can tailor drugs that are developing that. And GlaxoSmithKline, our major pharmaceutical partner, are interested in developing epigenetic drugs and epigenomics, but they can't do that outside of simple on-off kill, no-kill data that is run routinely in pharmaceuticals. And they need more and more disruptive technologies to better inform the disease process, to better inform the mechanism of the drugs. Okay, so that was one kind of thread of CEAT as we put together. And then thinking about, if we have new drugs, how do we get them at higher concentrations and target the delivery to those diseases using antibody drug conjugate technology. And we had a track record with ADC partners in Wales that we wanted to build through CEAT. And we're pleased to say that, we have ADC partners, particularly with Bivitrix, who you're going to hear about today, and their take on this bispecific approach of developing targeted drug delivery moieties. And again, in order to understand, how to develop, how to move, how to better select ADC candidates and therapeutics, bispecific candidates, they need disruptive technologies and new understanding. So we really synthesized and brought together a range of industry partners to provide platform technologies, develop applications for those technologies and disruptive applications within the pharmaceutical and targeted drug delivery markets. Okay, and these markets, they provided us with scientific questions, with technical challenges, and kind of demand-driven challenges. And from there, CEAT was born. We developed a 4-year funded project, well, initially 3-year project, which has now been extended to 4 years due to the success of the consortia collaboration. Our project worth is now just over 5 million, I believe. And under the banner of SMARTExpertise, we've really been able to push the boundaries of this collaboration. So what does, Julie mentioned effective collaboration, what does it look like? It looks that, for me, effective collaboration is that each of the partners gets to develop their R&D and gets to develop ideas together, test their biotechnology and develop their research agenda in cohort with each other, really. Okay, so in RBGO, we have, as I mentioned, advanced disease models, and that's a really nice partnership with our clinical partners. Axis Bio, one of the partners in CEAT, also has expertise in developing these models, in transferring these models to mouse and animal models and clinical translation. And they've really come in and helped us develop models for resistance, for example, and different co-culture models. We then interrogate those models using drugs, in this case from GSK. So the way the project is set up is in different technique platforms, divided broadly between epigenomics and ADC, and then within that, different platforms. So we take the GSK compound library, which is targeting epigenetic mechanisms that are altered in ovarian cancer disease. And in order to understand if those drugs are working, how are they working, and what the mechanism is, are they linked? Are the mechanisms linked to this mechanical environment, to the metastatic processes that I outlined in ovarian cancer? We develop applications and really challenging applications for disruptive technologies in the form of Chromatrap from Povey and BioAFM with Bruker. And I'm pleased to say that, that Bruker will be speaking later and telling you about their feeling and their understanding and how they see their interaction within the consortium. And then on the other side, in platform C, with the antibody drug conjugates, Swansea and RBGO and Stephen Dare have a long track record in developing antibody drug conjugates through one of our proprietary targets. We have selection process and a series of a pipeline of adding value and we took GE or Cytiva into that and used their BioCore technology to look at the binding kinetics and affinity of our antibody drug conjugates within our 3D models, both using recombinant proteins and stable on-off kinetics, and then cross-referencing that to fluorescence-based and efficacy-based readouts in our 3D advanced models. And moving that now to, a more bespoke by specific question led by our partners and by Victrix. Now, outside of that, we develop large data sets, as I said, and one of the platforms then is we have informatics challenges to bring these different evidence bases and data sets together to prove that novel disruptive technologies have applications, that they add value to these advanced therapeutic development programs. And I'm pleased to say that, all of the projects are doing well. So if we, I started off thinking about how do I extol the virtues of what we're trying to achieve, and really what we're trying to achieve is captured in our research highlights. So we've developed in vitro models, we've made very complex, advanced in vitro models, 2D, 3D, monoculture, co-culture. We can now reconstitute that tumor microenvironment and elements of that tumor microenvironment within our labs. We can characterize those models and really use them to take the compound library from GSK and identify epigenetic compounds that have high efficacy and are ready for clinical translation for ovarian cancer. So we've gone from 30 compounds down to 5, and 3 of those compounds show particular efficacy in our resistant phenotype models. While doing that, we've linked the mechanical phenotype of ovarian cancer and readouts to some of our cell and molecular biology, our more traditional readouts. And we're now using biomechanics to add value to understand the mechanism of our drugs. We can now take our gynecological cancers and characterize them and subtype them based on their epigenetic profiles and stratify those patients. And from the different individual targets, we can match back to our compound libraries and start to select, individual drugs for individual groups of patients, in line with stratified medicine. Similarly, we understand the proteomics, and we've developed how do we take the knowledge of the protein expression analysis for our different subtypes and develop antibody drug pipelines, including Cytiva and other novel technologies to increase the success rate of this preclinical development pipeline. Okay, so outside of our lead candidate, we have other lead ADC antigen targets that we've identified and are now developing through the project. We have new bispecific targets, both ones that we've designed within the project and those that were already proprietary to Victrix, which you'll hear about today, I'm sure. And again, it's not, if Wales wants to lead in this area, if Wales wants to be well thought of in advanced therapeutics, we have to be able to handle large sets of data. Welsh government are pushing the data strategy in Wales. We're tying back to patient data. We're tying back to, trying to make sure that we utilize the full spectrum of the data that we produce and trying to see if we can develop machine learning and artificial intelligence, algorithm-based analysis of that big data to really add value to both our motivation and to those of our partners. So I'm just coming to the end. I don't want to talk too long, but how are we getting on? We set out to try and prevent and treat and develop new ways of treating ovarian cancer, and I think that we're developing techniques to identify patients, as I said, and to develop new targeted therapies, both epigenetic and non-epigenetic drug treatments. We're advancing our epigenetic drugs using these cross-cutting technologies, and we're generating novel ADCs and bispecific questions ready and developing evidence bases for their selection for clinical development. So the big take-home messages for me really are that through industry collaboration, we're developing a highly skilled multidisciplinary team that we can be proud of, that Swansea University and Wales can be proud of, and that the guys coming through our team and in our industry partner team are all working collaboratively together. The lines of communication are strong, and we are pushing the boundaries in that regard. We're developing advanced disease models and we're further our understanding. And by furthering our understanding using these different technologies, we're giving them data and showing their application to new markets and including or increasing their market sector growth through these bespoke applications. So we're delivering added value to our industry partners in some pretty seismic market value sectors in terms of, from the ovarian cancer drug market to the ADC development market.